Hello there. Today we are going to be talking about photosynthesis. Photosynthesis takes place in plants. More specifically, it takes place in the chloroplast of the leaves. This enables the plants to use light energy to convert it into chemical energy, in which they break down carbon dioxide and water to sugar and oxygen. It is broken up into two stages: the light-dependent reactions and the light-independent reactions. The light-dependent stage takes place in the thylakoid of the chloroplast. Here we see an arrangement of two photosystems and an electron transport chain. The first photosystem, photosystem 2, absorbs light energy through its chlorophyll. It uses an antenna complex to funnel all the light energy to a reaction center. At this point, all the light energy is used to excite the electron to a higher quantum. The electron is donated by water, which leaves the plant as oxygen. With this excited electron, it can now be moved to photosystem 1, where more light energy is used to excite the electron to an even higher quantum, which is then captured by NADP to form NADPH. As the electron moves down the transport chain, hydrogen ions are pumped into the thylakoid, setting up a concentration gradient that will then be used to create ATP. This concludes the light-dependent stage, leaving the plant with two products, ATP and NADPH. These products will then fuel the Calvin cycle that takes place in the stroma.
Thank you, Jamie. Now I'm going to talk about the light-independent part. The Calvin cycle reduces CO2 to G3P through three steps. The first step is known as carbon fixation. Here, the enzyme Rubisco adds CO2 to a 5-carbon molecule, turning it into a 6-carbon molecule. This molecule now goes on to phase 2, reduction. Phase 2 uses ATP and NADPH to reduce the molecule to a 2G3P. This cycle happens 3 times, producing 6 G3P. 5 of the G3P are used to create the original 5-carbon molecule. This step is known as regeneration. The remaining G3P will be used to form glucose and other organic molecules.
Something very important to remember is the enzyme Rubisco, which drives the Calvin cycle. The enzyme was first produced back when the Earth's atmosphere was primarily carbon dioxide, but now the atmosphere contains more gases, most importantly oxygen, which competes with CO2 to bind to Rubisco. Because of this, a new species of plants have evolved known as the C4 plants. These plants are spatially separated and they have two types of cells. The mesophyll cells, which have an enzyme, PEP carboxylase, that will take a 3-carbon molecule and form malate, a 4-carbon molecule. This 4-carbon molecule will be used in the second cells, the bundle sheath cells, to produce carbon dioxide that will go into the Calvin cycle. Another type of plant are cam plants. An example of a cam plant would be a cactus. They are similar to C4 plants, but they use temporal separation. They have both enzymes, PEP carboxylase and Rubisco, and they capture carbon dioxide at night through their open stomata. When the stomata is closed during the day, the plant will use the Calvin cycle to reduce water loss.